Okay, let's just start. Hello, everyone, EUFRAS friends. My name is Edgars Zelme. I am a project manager for EUFRAS, and today it's my honor to be a moderator for this EUFRAS coffee break event. Today I'm replacing my colleague Anita, so don't be mistaken by the name. I'm not Anita. And today's topic, we have really interesting topic, breeding resilient grapevines from genes to IPM solutions. And as far as I know, for this coffee break event, we had more than 90 participants from 26 different countries registered. They are all not here, but I think they will see the presentation, see the video, and so on. So I think it's a clear sign that that this topic is relevant and interesting for most of us. So today we have three speakers, three inspiring speakers, titled speakers, if I can say. We have a Komlan Avia from the INRAE Research Center of Colmar, France. Then we have Li Teh from the University of Minnesota. And we have Giorgio Gambino from Institute of the Sustainable Plant Protection of the National Research Council of Italy. So if our speakers are ready, the floor is yours.
Thank you very much. So I have I'm in charge of starting.
Sorry,
this.
Coleman, one phrase.
Yes.
Sorry for disturbing. Just a reminder. Feel free to ask any questions. Write it in the chat or just raise your hands. Afterwards, I will read your question from the chat or just give you a word and then you can ask it yourself. Sorry, let's move on.
Thank you very much. So yeah, hello everyone. I'm Komlan Avia from INRAE Colmar, and welcome to this coffee break. I have the honor to start, and I will start by saying you probably already know that grapevine is a major economically important food crop worldwide, and especially in Europe, where it has, besides the economic importance, also cultural importance in several countries. Here you can see that European countries, by covering about 45% in terms of surface of the worldwide viticulture, they are in the top positions in the viticulture sector in the world. But you can see that either by in terms of surface or wine production, several major countries are emerging. Could be China, USA, Chile, or etc. So even though this production is very important in Europe, there is a very heavy competition worldwide. But we have now several challenges that the viticulture sector is facing. And if you are from a wine producing country, you already know that the situation is not that great the latest years. So one of those major challenges is that there is a heavy push either from the European Union or at national levels to reduce the use of plant protection products. And as you may know, those products are heavily used to protect vineyards against multiple pathogens, especially downy mildew and powdery mildew. And globally in Europe, we estimate that 2/3 of all fungicides used are applied in viticulture, while viticulture only represents about 4% of the agricultural area. The other bigger challenge is the climate change. And you also already know that overall, we now experience an increase in temperature with extreme events in several countries. And this also is linked with new pest and disease pressure, a higher frequency of abiotic stresses, and all this will have an impact on the wine quality and style, and also a shift in the wine growing areas. So in face of these challenges, one of the solutions we currently have in hand is breeding. And why breeding? Because we can develop new grapevine varieties that can carry resistance against those main diseases I talk about, and at the same time, that could be adapted to current environmental conditions. And today, the three speakers, myself and my two other colleagues, we will present through three different projects funded in Europe or in the US that are embarked in breeding programs aiming at providing solutions to wine growers in different countries. So GrapeBreed for IPM is the European Union Horizon or Europe project that I coordinate. VitisGenIII, it's American initiative that my colleague Dr. Li Teh will present. And last, Shield for Grapes is another Horizon Europe project that my colleague Giorgio Gambino will present. So I said one of the solutions we have in hand is breeding. But for breeding, what do we need? One of the first prerequisites for breeding is to understand the genetic determination of the agronomic and quality traits. So it means that we often need some information regarding how those traits are expressed in the genome. And this is done through what is called QTL mapping in biparental populations, or through genome-wide association studies in diversity collections. The results are generally the identification of genomic regions controlling the expression of our traits of interest, which we can then target with markers and carry out marker assisted selection. So this research also generates various omic data, could be classical molecular markers, expression data, proteomic data, or metabolomic data, okay? And marker assisted selection and genomic selection together with classical trait phenotyping form what we call conventional breeding. And marker assisted selection is now applied in routine for several crops worldwide, including grapevine. So this research can also continue toward the precise identification of and validation of the genes that are specifically involved in the control of the trait. And knowing this information, knowing the genes that are involved, allows us to carry out new genomic techniques-based breeding. You probably heard about CRISPR-Cas9, etc. And this is done, for example, by targeting a susceptibility genes to induce resistance in a specific varietal. Okay, so we have conventional breeding and we have NDT-based breeding. In conventional breeding, marker assisted selection is most of the time suitable for traits with simple genetic architecture, meaning that they have a simple determination, such as disease resistance, where we have one, two, or a few genes that are involved in the control of the traits. For grapevine, the common diseases we generally target are downy and powdery mildew, black rot, botrytis bunch rot, or trunk diseases. And typical breeding schemes in those programs are very long, starting generally with crosses between wild grapevine species and Vitis vinifera to integrate into vinifera background resistance factors that are naturally present in the genomes of those wild species. So you can see here that 15 years, that's currently the minimum or more, are usually required to complete those breeding schemes. But remember that such programs are very important because disease resistance not only allows to drastically reduce the use of plant protection products, but can also sometimes be the only solution to fight against some diseases when we don't have any chemical protection solutions. Okay. And so we have simple determination traits that are suitable for marker-assisted selection. And for traits that show complex genetic architectures, such as resistance to abiotic stresses, genomic selection will be more suitable. This approach involves preparing prediction models in what we call a reference population by linking phenotypic variation to molecular markers or globally omic data, and subsequently use those prediction models to predict phenotypic value in breeding populations at very early stages without waiting several years to be able to measure the threats in the vineyard. Okay, so genomic selection, therefore, has the potential of accelerating selection schemes, addressing complex threats, evaluating more plants, because since we can evaluate the plants at very early stages, we can get rid of those that are not interesting. Okay, so we save time, we save space. And last but not the least, to decrease the selection costs. Those programs have some costs and that are not negligible. So together with the plant material that we developed through those programs, our breeding project also have the objective to defining the best IPM strategies for farmers. So IPM means integrated pest management, and it is based on a diversity of pest management measures, including prevention, non-chemical control, best practices for optimizing pesticide efficiency, etc. And through IPM strategies, we want to reduce the reliance on pesticides, protect people's health and environment, and promote biodiversity. The projects we are representing today have the aim of providing farmers and advisors with best practice guide for disease-resistant variety cultivation, to promote alternative plant protection strategies based on decision support systems such as those solutions such as elicitors, resistance inducers, synthetic microbial communities, or green chemistry products. And because we are currently working on those solutions, those different projects will provide farmers and advisors with concrete solutions in a couple of years, and we suggest that we come back here next time and present the results regarding IPM strategies adapted to disease-resistant varieties in one or the future coffee break. Well, so now just a couple of words on the project that I'm coordinating, which is called Great Breed for IPM. We have 21 partners that are involved from 7 different European countries. We got 5 million from the European Commission, but also some funding from the Swiss government, and the project is bound for 4 years. Here you can see the different partners, from researcher to large producer or promoters of resistance or disease-resistant varieties. And quickly on the contents of the project, we have 3 main scientific work packages where we want to understand or better understand the molecular basis of disease resistance, to dissect the genetic determination of the threat of interest that we are interested in, and to embark on breeding programs in different European countries to make sure that after this project and in the longer term, farmers will have a solution to different disease-resistant varieties that can be adapted to their specific production context. We also have in this program a work package called co-design, where in different countries, we want to make sure that we gather people, stakeholders through the whole value chain and discuss to make sure that we agree on the barriers, but also the opportunities to make sure that we have a bigger deployment or a higher deployment of disease-resistant varieties in Europe. And as you can see here in Work Package 5, we are also working on developing the tools for IPM strategies that will go together with the plant material. As you know, all those programs, the outcome is those varieties I just talked about. And right now, there are numerous initiatives in Europe, but also in the United States and other countries in the world that are dedicated to creating those disease-resistant varieties. Those are resistant to disease, but not only, but also some of them also to different abiotic stresses, while simultaneously we try to investigate the pathogen population dynamics in different production contexts. You probably heard about some of those disease-resistant varieties. Some names are Artaban, Floreal, Artis, Sauvigny Gris, Aurélie, Fleurtail, Merlocorus, and there are more also to come. And if, as advisor or growers, you want to have information regarding those disease-resistant varieties, it's also called PIWI varieties in several countries. You can get information from nurseries in your country who can provide direct technical information regarding those varieties or put you into contact with the breeders, okay? Now we have some specific message for you. We need to retain that disease-resistant varieties or PIWI varieties must be considered fragile common goods as their resistance factors remain inherently vulnerable to breakdown, meaning that if you don't, by constructions, those resistant factors that are incorporated in those varieties as fragile can be broken down by the pathogens in the production context. So we have some measures to make sure that we infer some durability to those varieties, especially in long term. So to do that, they require a minimal yet consistent level of crop protection, at least 2 treatments around the flowering period, and they should be managed with the same care as the emblematic grapevine varieties. This is very important, okay? And to make sure that we have a bigger impact regarding the reduction of the use of plant protection products, we need a push from regulatory changes. And one of those pushes will be, for instance, to make sure that disease-resistant varieties are accepted in PDOs in different EU countries. So those varieties need to establish themselves because they are very new, very recent. They need some time to be recognized, to be known and fully understood by wine growers across different countries to reach the EU Green Deal objective. But right now, there are a lot of barriers, especially PDOs, which play a central role in structuring the wine industry in several countries. Fortunately, we start seeing, for instance, in France, since 2023, those disease-resistant varieties may be planted on a vineyard, up to 5% of a given vineyard, and their wines can account for up to 10% of a blend while still retaining the appellation status, which is very important because this can give more room to those varieties. And specifically in France, in the Champagne region, one of those varieties called Voltis is now directly integrated into Champagne PDO, which is a huge decision. And if a region or production region as Champagne can do such a thing, it means that other regions can also adapt such regulation, okay? Now, once those disease-resistant varieties are deployed, we need to track their durability because I told you that those resistant genes are fragile. And one of the way we do that is through one initiative that we have in France currently, and we are trying to extend to European level through the Grapebreed for IPM project. It's called Oscar Observatory. It's this observatory, it's a way to gather into one same observatory every grower that has a vineyard with disease-resistant varieties. And by getting information, we try to follow the effectiveness of the varieties, their behavior in production contexts, so that we can also track any emergence of sanitary issue, for instance, disease breakdown, resistance breakdown, and then alerts. And then researcher can also decide on measures that can be taken and be communicated to the growers. So this is very important. And I told you that there are several initiatives that are going on, not only in Europe, but also elsewhere, especially in the Europe. And now I'll pass the floor to my colleague, Dr. Li Teh, who can talk about a major initiative in the US, the VitisGen project. Please.
Thank you, Komlan. I hope all of you can hear me okay. My name is Li Teh. I'm a grape breeder at the University of Minnesota. I'm also very delighted to be representing the US grape research community in presenting to you some of the work that we've been doing over the past several years, particularly through the lens of VitisGen. So VitisGen is actually a national project initiative that has been funded on its third successive round right now. And so right now we're currently in VitisGen-III, and the project as a whole has really begun for about 14 years or so, spanning 20 research institutions, covering 65 scientists, and in total have been the funding amount has been just over $20 million. And really the core focus of VitisGen has been predominantly on powdery mildew itself, and the overarching goal has been to apply technological innovations to deliver breeding lines that have durable powdery mildew resistance, as well as in combination with high fruit quality, and also providing strategies to better manage existing vineyards. Next. And I'm sure most of you here are well aware of just how important powdery mildew it is when it comes to managing powdery mildew in the vineyards. But I'm here to emphasize the value proposition of powdery mildew resistance in the new variety itself. And this is a very simplistic economic model table that was done back in 2014 that basically shows the adoption rate of powdery mildew resistant vines and how that provides economic values depending on the speed of adoptions. So just as a quick example here, for example, if there's a 100% adoption rate of powdery mildew resistant vines that happens in 10 years, then we're looking at a $620 million in economic benefits, additional economic benefits. But if that is delayed by an additional 30 years, we're only looking at just the next one. If we're delayed by an additional 30 years, we're looking at a 40-year lag. That is, we're looking at only $250 million in economic benefits compared to $620 million. So that's a sizable difference by just delaying that an additional 30 years in the adoptions. And then the other thing is that, looking at the difference in these values, keep in mind that this model was projected on California raisin grapes. So if we think about it happening on the global landscape for wine grape, table grapes, and raisin grapes, the economic benefits in the adoptions is even much larger. And I hope this sends a quick message across the board, which is that when it comes to adopting powdery mildew resistant vines, it's important to embrace it holistically and also to be speedy in actually adopting them. So this is really provides an economic incentive to accelerate and increase adoptions. So next line. And one of the areas that, as I said earlier, that we've been focusing in the US is powdery mildew resistance. And that's really because the US is a center of diversity for powdery mildew pathogen, Erysiphus nectator or Ansonella nectator. And this figure on the right-hand side is showing the pathogen population diversity that you can see from the different clusters of bubbles over there. And that really corresponds to the US map on the left-hand side, looking at the geographic origin of some of these pathogen isolates. And so this really shows us that there's a lot of diversity that we can see in the US itself. And this presents an opportunity for us to stay ahead of the game by studying and understanding the genetics and understanding where hosts are resistant to the pathogen itself, so that we can really be staying ahead of the competition that we are competing with the pathogen itself. So we're really trying to provide growers with the solution. So this is why it's really important that we have been making a lot of progress in powdery mildew resistant discovery. Going to the next slide. And these are some of the results that have happened over the past several years in our discovery and our work for powdery mildew resistance breeding. You can see just a whole host of resistant powdery mildew resistant loci. I want to make 3 classifications here. The first is the texts that are in bold, and those are the loci that have strong resistance effect. And then the second one would be those fonts that are in regular fonts, and those are the ones that have moderate resistant effects. And finally, those fonts that are in smaller font size, and that you can see with the REN 10, SEN 1, and SEN 2, those are the ones that have a minor resistance effect. And really this, back, and you can see just a whole host of loci that have been detected coming from various different chromosomes across the 19 chromosomes in a genome. And you can also see that the different sources, so not just strictly related to rotundifolia, but also have since been uncovered on Vitis cinerea, Romanidae, Piasetzki, Astravallis, and Amurensis, and even also in vinifera itself. And I think some of you in the audience are aware of just the importance of understanding the race specificity. And of course, when we see that there's a yes for race specificity, we know that it's a gene for gene interaction, meaning that if we're adopting it by just one particular locus, there's a possibility that it can be broken down and overcome over time. But if the resistance itself is stacked, then the chance of being more durable is obviously goes up significantly. And then if one of the resistance itself is not race specific, it's a more broad spectrum, then it also has the possibility to be more durable over time. And I want you to take away a few key messages from this particular slide. Well, first of all, know that not all resistant loci are made equal, right? I've already showcased that there are some with strong effect, some with moderate effect, and some with minor effect. And second of all, it's important to know that the resistant locus itself, it can have different effects in different regions. So some of the locus that are important in one region may not be important in another region because it may have been broken down. For example, REN 3 is a fairly important resistant locus for me at the University of Minnesota. However, REN 3 has been shown to be broken down and overcome in the East Coast of the United States. So it's really no longer that important over there. And third of all, an important message I want you to take away is that I hope that as growers, as you're planting new vines, please try to get information from the nursery on how many resistant loci are in there. And then of the resistant loci that are there, what are there? Because it's important to just know if you have one or more than one, and then of the composition of the loci that have been reported, are they going to be significant? Are they going to be important for your region? So, and then the final point that I want to say here is that just because you are using or you're growing powdery mildew resistant vines does not mean that you're using this as a replacement for spraying any chemicals. We should really be thinking about using these powdery mildew resistant varieties in combination with a reduced spray schedule. So you're still going to spray, but you're not going to spray quite as much as traditionally. And this really allows us to protect a lot of the innovation and that many decades long effort that has been invested into breeding itself. And then next slide. And then in contrast to powdery mildew resistance breeding, downy mildew resistance breeding effort in the US is really pales in comparison because our focus has been predominantly on powdery mildew itself. And I'm sure most of you here in the audience are aware that the European group has been really the championing and pioneering in the downy mildew resistance discovery, particularly from group in France, Italy, and Germany. And so, but over the past couple of years, we've actually in the US have made significant progress. As you can see in the table, down in the table itself, we've made some discovery. And in fact, myself included right now, I have a PhD student that is working in my program, working on downy mildew resistance research project. And then I just want to also put a plug here that in Geneva, New York, they are establishing a new research vineyard of all RPVs in the Center of Pathogen Diversity. And this is a further extension of a gesture for international collaborations. So going to the next slide. So far I've talked about some of the research effort that's been done in the US, particularly through the lens of VitisGen successive series. And those were some of the scientific discoveries and findings. And now I want to shift gear to spend the remaining 3 slides to talk about some of the products itself that will be made available to the growers, because ultimately a lot of this discovery that's done through traditional breeding, we hope that it will become a product that growers can adopt and use. And so I procured 3 slides, one from 3 major public breeding programs in the US. So the first slide pertains to my program. You can see the first 2 varieties on the left-hand side. The 2 advanced selections wine grapes, and these are the ones that really potentially will be released in the next few years. And then on the right-hand side, you can see the 3 new seedless table grapes that my program just released this year. And so one of the things that you can see here are the different resistance loci that are represented here. And then very importantly, because Minnesota has really harsh winters, it's also important for me to breed cold tolerance as part of the portfolio itself. So you see the designation for zone 3 hardiness and zone 4 cold hardiness. So zone 3 would be considered more cold hardy than zone 4 cold hardiness. Next slide. And then the second slide that I managed to obtain was actually from a collaborator, Maddie Oravec. She's a new breeder at Cornell University. And here in these figures, you can see 5 grapes that are shown here, 4 wine grapes and also 1 juice grape. One of the things you can see that jumps immediately to you are just the number of resistant loci that are stacked in these advanced selections. So you can see run 1, run 2, and run 3. I mentioned earlier that run 3 has been shown to be broken down in the east coast of the US. And you can see that when run 3 are here, it's also here in tandem with run 1 and run 2. This really provides additional protection against powdery mildew itself. And then final slide. And these final slides come from great breeder Dr. Luis Garcia, Dr. Luis Diaz Garcia from University of California, Davis. And he has 5 new grape varieties over here that you can see here. And this actually re-emphasized a point that Komlan made earlier. Komlan stated just now that not all disease can be managed through chemicals and genetic resistance, right? And this is a really nice example. Pierce's disease is a disease that's caused by bacterial infection that currently there's no solutions other than trying to be, take final sanitary measure. But once the vines are infected with it, there's really nothing you can do much about it. And this is an area where genetic resistance are critical. And this is why you can see that of the 5 new varieties that are shown here, all 5 of them are highly resistant to Pierce's disease. So with that, go to the next slide. So I hope you get the chance to see, to catch a glimpse of some of the research work that has been done through the US, particularly through the lens of VitisGen. And I was showing a lot more from a traditional breeding perspective. But my colleague, Dr. Giorgio Gambino, would share with you some of the technological innovations and some of the pioneering innovations that he's been doing from his group, particularly through the lens of the another Horizon EU project, Shield for Grapes. So with that, Giorgio, please take it away.
Thank you very much, Li. Good morning, everyone. I'm Giorgio Gambino, researcher at the National Research Council of Italy, and I'm the coordinator of this last project presented this morning, Shield for Grapes. It's a European Horizon project. The consortium is composed by 60 different partners from 7 countries. And is a consortium, multi-actor consortium with research center, university, wine growers, nursery producers, all the viticultural system involved in main viticultural countries of Europe, and is also externally supported by the OIV. Next, the project, the main objective of the project is to develop a new grapevine variety to improve tolerance to resistance to fungal disease, in particular powdery downy mildew and grape round diseases. And in addition, to apply IPM strategy specific for this kind of new genotypes. The approach used in the project will be a traditional conventional breeding, the marker-restricted selection already well presented by my previous speaker. And an interesting important point for our project is the application of new genomic techniques, in particular the cisgenesis and genome editing, to technique not GMO, don't produce plants considered GMO. And another interesting opportunity, the somaclonal variation, a regeneration system useful to produce mutation in the traditional varieties. Also, in this case, this approach is not a GMO approach. Some example, for example, the cisgenesis. The cisgenesis is in comparison to traditional transgenesis and GMO production. In cisgenesis, we have the transfer of a complete gene between variety of the same species or interspecific species. This process mirror in some way the transfer of gene what happened in the traditional breedings. This is a difference from GMO production and transgenesis, because in the GMO we have the transfer of gene from every kind of organism, in bacterial, animal, in plants, and vice versa. Another difference important from GMO is the absence of DNA not belonging to VitisGenIII, which without the sequence from antibiotic or herbicide resistance. And inside the project, we start to try to transfer some interesting gene from a Chinese Vitis quinquangularis, showing tolerance, a good interesting tolerance to powdery mildew and to other abiotic stresses. I try to transfer this some interesting gene in the traditional varieties. Other approach of consider new genomic techniques is the genome editing. It's very powerful process technique developed, discovered more or less 12 years ago, awarded by a Nobel Prize in Chemistry in 2020. It's a very important techniques. And it's possible with this application of this technique, the target induce mutation in specific point inside a genome of a plant, of an organism. In the case of wine, it's possible to produce variants of traditional varieties with inducing mutation very similar to the natural mutation which happen in the vineyard, in natural environments. And another interesting point is that no new DNA sequence was inserted in this in the edited plants. Some example of traditional variety improved is the Chardonnay, very important international variety, in which we have induced mutation in this gene, MLO genes, is a susceptibility gene for the susceptibility to powdery mildew. This mutation induced the very strong resistant, strong tolerance to the powdery mildew. And other plants with the mutation in the other gene, MPR3, we obtain plants with a good tolerance to both the disease, powdery and downy mildew. This is very interesting new genotypes at the moment in conserved in greenhouse. Another example is another Vitis vinifera variety, Nebbiolo, is a very important variety especially for the northwestern Italy, because was used for the production of very important high quality wine like Barolo and Barbaresco, known more or less worldwide. And in this case, we have target another gene, this microRNA4A2, and useful to induce a broad spectrum of tolerance to different pathogens. And in the next few weeks, we will check if this new plant is effectively more tolerant to some pathogens. An important question is that these NGT plants are ready for the vineyards. The first point in the coming years and coming months, a very high number of traditional variety will be improved through the genome editing. In addition to Chardonnay and Nebbiolo, probably in a few months we have the consortium of Komlan and other research group in Europe, outside Europe, produce several new edited plants from the traditional variety. Anyway, we have a big problem for the restriction in field for Europe, because in Europe, I think only in Europe today, the NGT plants are considered the GMO under the same restriction, the same legislation. Even if from the scientific point of view, NGT plant is not GMO, from the legislation Europe is today is compared to GMO. Anyway, Europe moved in the last 2-3 years, try to change this legislation. There is several discussions between Commission, Parliament, European Council. And now the European Union is now considering introduction of to differentiate the NGT plants to the traditional GMO. In this way, because NGT plants don't have any strong problems from the point of view, environmental health point of view, like the GMO. Anyway, this change of legislation is very important, because the future of this kind of approach in Europe is linked to this change of this legislation. The last approach for change, to modify the traditional variety, is the somaclonal variation. Only some few definition. Somatic embryogenesis means the development of embryos from plant somatic tissues. It's a characteristic typical of plant. It's not from animal, for example. This way, from a single cell, from a plant, is possible to regenerate a plant identical to the traditional, to the mother plants, to the traditional varieties. The somaclones, we mean vine plants, obtained to these new techniques. And in general, the somaclonal variability is a variability resulting from mutation, genetic or epigenetic mutation. And it should be very useful for developing new variants of traditional varieties, more tolerant to environmental conditions. Next. In this slide, we can show a very schematic overview of the process for producing somaclone from the beginning of the in vitro culture of flowers. In vitro culture to produce somatic embryogenic calyx, somatic embryos, and new plants in vitro condition. After, from the in vitro condition, to move to the greenhouse and come back to the vineyards. The complete process more or less need 2 years, 1 year to the in vitro culture, from the flowers to the production of new somaclone in vitro condition, and another year from move to the in vitro condition to the greenhouse, and after to the vineyards. And the somaclonal variation should be interesting to produce new mutants through the in vitro application of selective pressure in order to regenerate, induce the regeneration of somaclone more tolerant to some biotic or abiotic stress. This selective pressure should be, for example, a resistance inducer for increased tolerance to biotic stress, or other molecular compound like polyethylene glycol to simulate the water shortage in vitro condition. And one example, we can move. One example of the new somaclones obtained will be, for example, the somaclone from Bragata Rosa is a local variety of Italy, a very minor variety with not very important from the economic point of view, but it's important because it's a very responsive variety for this kind of regeneration. Anyway, we obtain different somaclone with an interesting reduction of the disease severity after infection with Plasmodium viticulum. Another example is and again, the Nebbiolo variety, in which in this way we have regenerate somaclone more tolerant to drought. After 2 or 3 weeks of water shortage in greenhouse condition, we see some somaclone show a very interesting response, physiological response, with increase in stomatal contents or less negative water potential, which means an interesting tolerance to the drought in comparison of the original mother plants. Another example is the rootstock 100 at Richter. This kind of activity should be useful also not only for vinifera, but also for the other species. In the case, the commercial rootstock 100 at Richter, we have obtained soma clones, in this case in an open field condition. We have also in this case some interesting soma clones more tolerant to the original mother plants. At the end, we can move. The soma clones is ready, is a system ready for the vineyards, because we have started with the first field in Italy in 2024 with several soma clones from international variety area and Italian variety. After 2 years in this season, we have collected some interesting data from some of these soma clones, in complex more 600 different soma clones. And in this winter, we start to collect some wood for start to the grafting with the commercial rootstock or new rootstock 100 Richter. And the next year, we move to the commercial vineyard with this new soma clone grafted. The next year is important because we obtain new soma clones from not only from international variety, but also from variety from different location in Europe, from Portugal, local variety from Portugal, Spain, and also in Hungary. In this way, it's possible to cover with this project more genotype, also local genotype from different countries. Next. In this final table, I'd like to summarize the difference of these three approaches of genetic improvement talk today in these three talk. Comparing the disease-resistant variety produced from traditional breeding and marker-restricted selection, new genomic techniques, and soma clones. It's important to remember that the disease-resistant variety is a new variety, which can lose some typical characteristic of traditional variety. While a new genomic technique, the soma clone, will produce a variant of the traditional variety. It's possible to maintain the identity of the wine, the typical wine. For disease-resistant varieties, the breeding cycle is a little bit long, 10, 50 years. For the other techniques, the time is a little bit short because for more or less 2 years to produce the plants and to move outside the vitro condition. After 4 or 5 years to try to analyze the plants in open field condition. Disease-resistant variety is very specific against some pathogens and generally induce a very strong level of tolerance of resistance, as we have seen before in the presentation. For the soma clone and GT plants, probably it is should be less specific to specific pathogens or disease, but probably induce a lower level of tolerance of resistance. But we will see probably in the next years. And another interesting point is the disease-resistant variety and new genomic technique need to know exactly the source of resistance, of tolerance, the gene involved in the tolerance in the susceptibility, to transfer the gene or to modify the gene. The soma clone don't need to know exactly the genome of the plants. And the last point, but it's not less important, is the regulatory framework of this different kind of new breeding. For disease-resistant variety, Komlan already talked about the restriction about in PDO region from some countries. For NGT plants, we have a strong problem from the European legislation. And soma clone is already more ready approach because is considered natural variants of traditional variety, is free to plants in vineyards, also in DPO regions. There is the last one slide with a general take-home message for this morning. I think it's important to remember that I understand that European viticulture need to try to apply the genetic improvement also in grapevine, because without the genetic improvements, I think it's possible, we think it's possible to meet the objective of the European Green Deal. Because grapevine is one of the few crops where the genetic improvement has not had a significant impact on the history, because the traditional variety cultivated today was selected hundreds of years ago with under a completely different climatic condition and pathogen pressure. It's completely different of the situation of today. There is not a best approach for grapevine genetic improvement, because every approach should be used for some situation, some sociocultural background. Because remember, disease-resistant varieties, new genotypes, restriction in PDO, but not all European countries change the legislation. For example, in France, we have some example of the Champagne region. It's possible to introduce one new variety. For example, in Italy, we have many varieties registered in the National Register of Variety, but today it's not possible to insert, to cultivate in PDO. But it should be very interesting for the organic viticulture, this disease-resistant variety. The other side, the NGT is traditional variety, but strong problem for GMO legislation. In general, another point important, there will never be a super wine, because completely tolerant to all disease, all environmental condition. As we see before, even the disease-resistant variety showing a very strong tolerance to powder downy mildew needs some treatments. At the same time, a variety may be useful because tolerant to powder downy mildew, xylella, or other disease should be not so adapted to environmental condition, maybe susceptible to drought, for example. This is the super vine is a dream. In the end, the last point, for a resilient viticulture, we need not only the genetic improvement, but we need also an effective vineyard management and effective IPM strategy. All the three projects presented today, we focus on IPM strategies, focus on the specific new genome. And I think this aspect will be developed before the end of this project. In 1 or 2 years, we have some data interesting also for these new IPM strategies adapted for these new genotypes. I'd like to thank everyone. In this last slide, you can see the link for the website of the three projects for the update. I thank you, the participant. I thank all the three speakers, Komlan and Sun Li, and we are ready for questions.
Yeah, thank you, all three speakers. It was a huge amount of information for me. I'm still processing all of it. Like, yeah, really huge job there. We have a question from Sarah. I know she already got the answer, but the question is for the Professor Teh. Are there forecast models to schedule the reduced number of powdery mildew treatments on resistant varieties?
Yeah, so this work is conducted by Dr. Katie Goh at Cornell University, so I really can't speak on her behalf. I believe she's currently in the data collection stage, so there should be more information coming out in the next couple of years. But my suggestion is that if you reach out to her directly, you could probably be able to get some type of unpublished information and see whether or not that would guide your perception. And I think another important thing is also to realize that all these forecasting models, they really are concentrated on some of the common varieties first. And so it may or may not be directly relevant to you, but at least it provides a framework for understanding how you think about a reduced spray when it comes to a disease-resistant variety.
Yeah, thanks a lot. Do we have any other question from the audience who wants to talk now? We have a last minute for the questions. If you have question, please raise your hand and go ahead. We had some questions more the chat, but you already did that. Okay, I think the information is really interesting, but the amount of the information is so huge, and people are still processing it. Yeah, because it's a huge topic, and then we went really deep in that, and Yeah. As I can see, we don't have any more questions now. For audience, if you have any questions further, you can contact our speaker directly, and also you will get the presentation. What I want to say, thank you all for being with us in this event. I really hope that you got some kind of insights or some kind of new thoughts or new knowledges or new inspiration from this all. And it was in some ways useful to you. Please remember that the recording of this session will be available on EUFRA's YouTube channel. If you, in case you would like to use within your colleagues or somehow use it in your job, please feel free to do it. And one more thing. We look forward to seeing you at our next EUFRAS coffee break, which will happen in 30th of October, and it will be dedicated a little bit more specifically to young advisors. So yeah, feel free to join, and we're looking forward to seeing you all.